Hello, folks. This is Dr. Jenkins, and we are here to go through the amino acid chromatography lab. I'm very excited about this one. Very cool. So what we're going to do is we're going to do a little introduction about what chromatography is, and then I'm going to walk you step by step through the process, and then we have some results for you to look at. And then you'll be able to record those results for your online lab. Okay, folks, so we're studying proteins. And in here is a nice review about the functions of protein. I want you to remember that everyone's going to remember structure. That's the easy one, but not just muscles. We rely on amino acids for skin, hair, nails, ligaments, tendons. Those are all very important for an athlete. Number 2 function of protein, as I've been saying again and again, is that protein is needed to form important chemicals in the body. Almost every important chemical that you think of is made up of proteins. Couldn't live without them. Hormones, enzymes, antibodies, neurotransmitters, hemoglobin, which is what allows us to transport oxygen in the bloodstream, all made up of protein. And of course, we're only going to use protein for energy in very small amounts. Instead, we want 85 to 90% of all of our cellular energy to come from carbs and fats. Okay. A quick review about the amino acids. We know that there are 20 of them in total, and every amino acid is some combination of some of those 20. Here's the general structure, and we'll be reminded that what makes each amino acid different is the side chain. The remaining parts are the same. It's the side chain that makes leucine different from valine, for example. Make sure you review of the 20 total amino acids. They're broken down into ones that our bodies can make. The ones that our bodies can make are considered non-essential. 11 out of the 20, over half, our body can make for us. Versus the essential amino acids, the remaining 9, that our body cannot make for us, and therefore, we must get through our diet. All right, continuing on, here is a listing of the essential and non-essential. This lab, we're going to be focusing on branched chain amino acids. You've heard of them, BCAAs. What are they? Is their supplementation the magic pill? Well, you can already guess my answer to the second part of that. No. Let's talk about what they are. Branched chain amino acids, leucine, valine, and isoleucine. So branched chain amino acids are those 3. They're a collection of 3 essential amino acids. And what's important about these, they are digested more quickly than many other amino acids. And here's what I really want to point out. These 3, leucine, isoleucine, and valine, they play a role. These are the amino acids that help us in muscle protein synthesis, which means these are the ones that help us build muscle. Can I say this again? These are the 3 essential amino acids that help us build muscle. Not every amino acid is used for muscle. Do you hear me? Loud and clear. Not every amino acid is used for muscle. So therefore, the thinking that taking in every other amino acid is going to help me build muscle, not necessarily. Remember, a protein has many other functions, or can go to structure of other things. The 3 that we rely on to build muscle are these 3. Leucine, isoleucine, and valine. They're also important to athletes because they can be digested more quickly. Now, do we need supplementation? Of course not, because these branched chain amino acids are commonly found in food. You're going to get a lot from meat, poultry, seafood, dairy, nuts, seeds. The ratio that we like is 2 to 1 to 1, where it's 2 parts leucine to every 1 part isoleucine and 1 part valine. We can get a lot. Look, 1 serving of chicken breast, only 3.5 ounces. Usually we're going to have more than that, contain a whole bunch. And they're in the proper ratio where it's the most leucine, smaller amounts of isoleucine and valine. But despite the fact that we get a lot of branched-chain amino acids from food, not just meat, seafood, look, dairy, nuts, seeds, you don't have to necessarily have meat. Despite this, many athletes choose to intake a branched-chain amino acid supplement. May I remind you, supplements are not closely regulated, so we're not even assured that that supplement powder contains what it says it does. Okay, so we've done the first part of this lab, review the structure and function. Now we're going to talk about chromatography. And the whole fun of this lab, folks, is we're going to put a branched-chain amino acid supplement powder to the test. I have done this in the lab. I've taken a branched-chain amino acid supplement, and we're going to see, does it contain what it says it contains? All right. Make sure you review the basics of chromatography. Chromatography is a separation technique. In the world of chemistry, every chemist knows all about chromatography. In its simplistic form, it's a separation technique. And I give you some examples. A sports drink. We could utilize chromatography to separate the sugar and the electrolytes from the water portion of a sports drink. So chromatography, even though it sounds fancy, is quite simply a separation technique. What are some applications to athletes? A sports drink. What's in a sports drink? Well, mostly water. It's probably going to have some sugar, some glucose, and amino acids. So chromatography would be a way in the lab to separate the sugar from the electrolytes from the water. Another example, different method of chromatography, but again, chromatography just means separation. Another example would be blood testing for certain drugs. Athletes, if they're in a professional sports arena, organization that requires drug testing, guess what? The drug testing is a form of chromatography, where we could separate the blood components, just the regular blood cells and plasma, from particular drugs that we're looking for, or drug metabolites. Okay, I'm going to let you read through more of the specifics. I'll be very general. If we were doing a more chemistry-focused class, we would talk about the stationary phase versus the mobile phase. I'm not as worried about that, but I do just want to talk about, in simplistic form, how we do it. So we have a beaker, check out the beaker, and we have a paper that's coated with a chemical. We put that rectangular piece of paper in a little bit of water. On that piece of paper is a sample to be tested. So we put this rectangular piece of paper with some kind of chemical on it. So let's say we have a chemical here and a chemical here. Over time, this water is going to move up through the paper. You know, if you put a piece of paper, even just the tip of the paper in water, the water will kind of osmose or diffuse up the paper. So what happens is, as that water starts to move up the paper, it takes a special solvent here with it. And as that water moves up the piece of paper, it can take a piece of the sample with it. So this would show positive for this chemical, and this chemical. Even if you don't understand the chemistry part, please know chromatography is a separation technique. Simply, we put something in a beaker, we fill it with a little bit of a special solution, and we put a piece of paper on there, and that the fluid from this water portion is going to rise up the paper. And based off of chemical reactions, it may show certain things as positive on the paper. Okay. After you do that, you have to take that piece of paper out and use a blow dryer, literally, to dry it and add this other spray that's going to cause it to change color. That's the developer. I'm not as worried about that. But let's get to what I'd like you to do. So in addition to being able to answer some general questions about amino acids, branched chain amino acids, essential versus non-essential amino acids, and what chromatography is, your first assignment here as part of this is to do a sample reading. So if you recall, one of the examples of how chromatography can be used for athletes is drug testing. So here, folks, is a sample drug test. So here is our blood sample from the athlete. Over here are the knowns. So imagine this whole rectangle is that piece of paper that we put into that beaker, and we allowed the fluid to travel up the paper. And as the fluid traveled up the paper, it took certain chemicals with it that can show as dots. So here's how you would read this sample. Here we have our knowns. So here's known number 1 for growth hormone. Growth hormone in this particular test would show as a red dot here. Then we have our second known. So we purposely have growth hormone put on the paper, and it shows as that red dot. Our second known would be anabolic steroids. It's going to show as this yellow dot. So those are our knowns. Over here is our unknown. So your job is to compare. Does the unknown have a yellow dot similar to this one? If it does, it's positive for steroids. If it doesn't, negative for steroids. Does this unknown sample, if you follow along horizontally, does it have a red dot similar to this one? If it does, then it's positive for growth hormone. So your job is to compare the unknown dots to our knowns. And if any of them are similar across, it's positive. So on your summary sheet that you hand in to me, you're going to record the results here. Based on this rectangular result, what did our unknown blood sample test positive for? All right, let's go on to part 2. Part 2 is putting a supplement to the test. So in the lab, I had a branched chain amino acid supplement powder. In running this in the lab, as you can see the instructions say, you would have made your own rectangle out of paper, and we would have drawn lines on the paper and labeled them. This is what's going to go into the beaker, right? And again, we're going to be looking for something like this, okay? You'll see. Here, the branched chain amino acid powder. I don't have a picture of the label, unfortunately, but the supplement says that it contained all 3 branched chain amino acids. Remember, What are the branched chain amino acids? Leucine, isoleucine, and valine. These are the 3 branched chain amino acids. They're digested and absorbed more quickly than other amino acids. And even more importantly, these are 3 amino acids that we use to build muscle. This is what the label said. So what are we going to test it for? In this lab, we're testing for, so we took the powder and we're going to test for leucine, isoleucine, and valine. But we also put 2 wild cards in there, glutamic acid and phenylalanine. Phenylalanine. Let me say that again. Phenylalanine. So we're testing for a total of 5 amino acids. If the label on the powder is correct, it will test positive for leucine, isoleucine, and valine, but none others. So we will see, will it contain the 3 that it says it does? And then I also threw in 2 other ones that the label says it does not contain. And actually, rightfully so, these are 2 other amino acids that do not play a role in muscle protein synthesis, in building muscle. And these are not branched chain amino acids. So we have a way to control and see if it tests positive for what it should, and then we just threw some other ones on there. Okay. So we made those strips. Remember you made those strips? Put the samples on there. Put it in a beaker with some fluid. All right. What I want to point out, though, before I show you the results, I want to go back for a moment. When we're doing chromatography like this, our results are qualitative. Qualitative means that it only shows the presence of something. It is not quantitative. You know these words, I'm sure. When I think of quantitative, I see an N and I think about a number. Something that's quantitative gives us a number. If this were quantitative, it would not only show us whether or not something is present, it would also tell us the amount. But the form of chromatography that we're talking about is not quantitative. It doesn't tell us how much in numerical value. It doesn't tell us how much there is of something. It can only tell us qualitative results. It can only tell us whether or not something is present. All right, so remember, we are testing for 5 amino acids, the 3 branched chain amino acids, and we threw 2 others on there also. The label said that it only has leucine, isoleucine, and valine. So we'll see if it in fact contains what it says it should. And then we threw on 2 others to see if there are some things in there that maybe shouldn't. These are, here's going to be where you record your results. This is our supplement. And out of those 5, you're going to write down what it tested positive for. These are real results. And by the way, we only threw in phenylalanine and glutamic acid just because we happened to have them in the lab. There wasn't really a rhyme or reason. We just happened to have phenylalanine and glutamic acid in our lab for controls. So it wasn't really thought of in advance. We just happened to have them. All right, drum roll, please. Are you ready for your results? I can see you're excited. Here they are. So here is our rectangular piece of paper. Oh, excuse me. I think I have to sneeze again. Hold on. My apologies. We're going to read these one at a time. So here in green is the dot that we did for the supplement powder. Here was the leucine control. And here was the isoleucine control. So just like we did before, here is the leucine control. Here is the isoleucine control. Now we look at the branched chain amino acid powder. If there's a dot at the same level as these two, we would say it's positive for leucine and isoleucine. What about valine, you said? Well, we did a separate sheet of paper just because of space. Valine shows positive as a dot right here. So the branched chain amino acid powder, here's where it showed a dot. If there's a match horizontally, it's positive. Lastly, here were the two wild cards that we threw in there. Again, here's the branched chain amino acid powder, and it showed here phenylalanine. That one showed here. Was the branched chain amino acid positive for phenylalanine? And then down here is the glutamic acid. So the branched chain amino acid powder is only positive for things that were in line with it. So based on this, I'll erase this so you can see it again. And if you have any questions, you can email me to ask. So from this, we can see what was positive in our branched chain amino acid supplement powder and what was negative. All right, folks, I look forward to seeing your results. I wanted to show you this simple separation technique. And any questions, let me know.